Imagine this. Your DNA, the unique handbook that makes you who you are, isn't entirely your own. Around 8% of your genetic code is made up of ancient viral fragments, leftovers from infections that struck our primate ancestors millions of years ago. What once posed a threat now lives on inside us, not as an enemy, but perhaps as a silent partner in our story. If you enjoy uncovering hidden secrets of our DNA, don't forget to subscribe so you won't miss the next discovery. In this piece, we'll explore the mysterious world of jumping genes, DNA sequences long dismissed as junk, but now revealed as crucial players in our development, our evolution, and possibly even our future. When scientists first mapped the human genome, they expected to find a neat blueprint filled with instructions. Instead, nearly half of our DNA looked like clutter, repetitive stretches with no obvious purpose. These mysterious pieces are known as transposable elements, or TES, often nicknamed jumping genes because of their strange ability to copy and insert themselves into new spots across the genome. For decades, TEs were dismissed as useless baggage, junk DNA left over from ancient accidents. But recent research paints a very different picture. Far from being waste, these mobile sequences may help orchestrate when and how our genes are switched on or off. In other words, they act like hidden control panels within our cells. This shift in perspective is dramatic. What we once thought of as evolutionary noise may actually be one of the driving forces that shaped our species. By moving, mutating, and even reprogramming genes, TEs could have given our ancestors the flexibility to adapt, survive, and eventually become human. How did viral code end up embedded in our DNA in the first place? The answer lies in infections that took place millions of years ago. When our primate ancestors were exposed to viruses, some of those viruses did more than make them sick. They left permanent marks. Bits of viral genetic material slipped into the host's chromosomes and, instead of disappearing, were copied and passed down from generation to generation. Today, those fragments remain as silent echoes of long-forgotten battles. For the most part, these sequences are harmless, kept in check by protective mechanisms within our genome. But they are far from irrelevant. Some viral remnants, known as endogenous retroviruses, ERVs, have been co-opted by evolution. Rather than acting like parasites, they became part of us, influencing gene activity, helping regulate cell development, and perhaps even shaping key aspects of reproduction and immunity. Of course, not every viral insertion turned out to be beneficial. Certain transposable elements have been linked to cancer and other diseases when they reactivate in the wrong context. Scientists are now investigating whether silencing or redirecting these rogue elements could open new doors for treatment. In short, the viral past written into our DNA is a double-edged sword, a potential threat, but also a surprising source of innovation. Although scientists have known about transposable elements for decades, their repetitive nature makes them notoriously difficult to study. Sorting them into families and subfamilies is like trying to piece together a puzzle made almost entirely of identical fragments. For years, this complexity kept their true roles hidden. A recent international study has begun to change that picture. By sequencing and reclassifying a group of elements known as MER11, researchers discovered 4 new subfamilies that had previously gone unnoticed. One of these, called MER11G4, stood out in a striking way. It showed a powerful ability to activate genes in human stem cells and early-stage neural cells. Why does this matter? Because stem cells and developing brain cells are where much of our identity as humans begins. The fact that a viral remnant could switch on key genes at such an early stage suggests that it plays a critical role in shaping development. These findings don't just fill in missing pieces of our genetic puzzle, they hint that ancient viral DNA actively influenced the emergence of traits that make us human. In other words, what was once dismissed as junk may turn out to be one of evolution's most creative tools. If transposable elements can regulate genes and influence early development, then it's no surprise they may also have shaped the course of human evolution. By comparing TE activity across species, researchers are beginning to trace how these sequences helped define what makes us distinct from other primates. In the recent MER-11 study, scientists examined genomes from humans, chimpanzees, and macaques. They found that while the same families of TEs exist in all three species, their activity and evolutionary paths diverged. Some subfamilies became more active in humans, while others faded in our relatives. These differences may have nudged our genomes toward unique developmental outcomes, changes in brain growth, immune response, or other traits that separate us from our closest cousins. To grasp the scale of TE influence, consider a very different creature, the Greenland shark. Its genome is made up of more than 70% jumping genes compared to less than 50% in humans. While sharks and primates couldn't be more different, the comparison underscores a crucial point. The balance and behavior of TEs play a central role in shaping how species evolve over millions of years. By studying these patterns, scientists hope to uncover not just what makes us human, but also why we develop diseases that other animals escape. Beyond their evolutionary story, transposable elements may hold practical lessons for modern medicine. Because some of these sequences can switch genes on or off, researchers are exploring whether targeting them could help treat diseases or even unlock new forms of gene therapy. One recent study suggested that silencing specific TEs could make certain cancer treatments more effective. If rogue elements drive tumors by activating harmful genes, shutting them down might give patients a better chance at recovery. On the flip side, other TEs may be harnessed as tools, deliberately reprogrammed to turn on beneficial genes or repair damaged DNA. The promise doesn't end with therapy. By mapping which TEs are active in different tissues, scientists may uncover why some diseases affect humans but not other animals. For example, if a TE regulates immune genes differently in people than in primates or rodents, that difference could explain why certain conditions are uniquely human. In short, studying these viral fossils isn't just about understanding our past. It's about equipping ourselves for the future. Every discovery brings us closer to new treatments, deeper insights into disease, and perhaps the ability to rewrite the very code that defines us. What once looked like genetic junk now emerges as a hidden engine of innovation. Ancient viruses woven into our DNA may have helped shape our development, our evolution, and even our vulnerabilities to disease. By unlocking their secrets, we gain more than history lessons. We gain the tools to understand and perhaps redesign what it truly means to be human. If you found this story intriguing, consider subscribing and share it with others who love exploring the hidden side of science.